Dr. Krakowiak will help me monitor the chat a bit, and we will go ahead and get started.
I'm still checking a little bit of the previous chat, so Jennifer, maybe you can see the bottom and I'll do the middle and we'll meet somewhere.
Got it.
Okay. Oops. Here we go. Okay. So this talk is going to be a smaller portion of a larger talk that I gave last month over COVID-19. I actually took only the last 2 sections of that talk and kind of expanded them into 3 sessions for this one. I'm going to be covering mainly the tests, like when you get tested for COVID, what does that mean? And the treatments that are available right now and the race to develop a vaccine. So as of a couple of days ago, we had roughly 14 million cases worldwide, over 600,000 deaths. On July 18th, which was the day that I updated this slide last, more than 260,000 COVID-19 cases were reported to the World Health Organization. That's a record. And I think that something like 11 of the previous 20 days had been records at that point. So clearly we are not getting this under control. And the United States has a number of hotspots in it. So this is not something that went away with the summer the way some people were hoping or predicting. This is going to be your first Pear Deck question. So drag that icon. Tell me how you're feeling about the pandemic and its impact on your life right now. Okay, we see it kind of all over the board. That's what I'd be expecting. I kind of feel, to me, I'm sort of in this region right here. Like, I feel like I've been fairly lucky so far, but I definitely know people down here as well. And so I do want to encourage you, particularly if you feel like you're on this end of the spectrum, please reach out, please find a support network, please talk to people, don't isolate yourself. This is hard, we know it's hard, and we want to make sure that we're all taking care of each other. This is going to be the table of contents for today. I'm going to start out with the tests, then go on to the treatments, and then talk about the race towards that vaccine, and especially what does that word race mean, because I think that the accelerated timeline has a lot of people kind of nervous, so I'll be talking to exactly what that acceleration looks like. But we'll start out with the tests. There are, I feel like, I'm not really sure what's going on here, but none of my transitions are set up.
It's okay. Sometimes in Pear Deck, it's an issue.
Okay, like all of these were supposed, like I'm supposed to be able to control these and they pop up one at a time. So
add on that we need to fix with that, but no.
It might be an issue with that. Okay. So anyway, there's 2 different kinds of tests for COVID-19. There is, are you infected now? Or were you infected previously? And some people just call this a COVID-19 test, and some people call this an antibody test, but they are different and they're answering different questions. So one is an RT-PCR test. Usually there are a couple of others now, also called a viral test. This is where you either get a nasal swab. If you guys want to, I don't know, drop in the chat if you've had a nasal swab, what was it like? Or if you have been tested for a current infection, how was that? When I had a test, my church had like a pop-up testing site where you could go and be tested for free. Anybody, like you didn't have to have symptoms or anything like that. So my husband and I went and we had to swab our own noses, and that swab, it goes all the way up there. Like they say, poke it up to where like your eyes are watering and then back off a little bit. That's what the person told me to do. So it's kind of uncomfortable. My eyes watered a lot. My husband coughed a lot. We both wound up being negative, although it took us 2 weeks to get that information. But this shows a current infection or potentially a very recent infection. The very recent being if there's a little bit of viral RNA that's left, even if all of the replicating viruses are gone. The other possibility is, were you infected previously? That is an antibody test. I had a cousin text me a few months ago and say that they were going to have a friend come stay, and he'd had a negative test. And so, was it safe? And I said, well, was it an antibody test or was it a PCR test? And she said, does it matter? And I said, yes, it matters, because they test different things. So the antibody test or serological test, this is looking for specific antibodies in a blood sample. And it may not actually show a current infection. One can get an antibody test and have it come back negative, and that could mean that they've never been infected, or that they've only been recently infected, because it takes 3 days, 4 days for those antibodies to start showing up in the blood after the infection has already started. So those are those two different tests. This one's looking for the viral RNA for a current infection. This one is looking for antibodies in the blood that would have been made due to a previous infection. Let's look at the RT-PCR a little bit closer. This is that question, are you infected now? SARS-CoV-2, this is the name of the virus. So when you hear me talk about this coronavirus, sometimes I'll say the virus or SARS-CoV-2. It has an RNA genome. And so what reverse transcription does is it takes the, we've got like a normal transcription process is DNA being used as a template to make RNA. Reverse transcription is that opposite direction. RNA being used as a template to make DNA. So in RT-PCR, reverse transcription is used. There's an enzyme called reverse transcriptase that is used to make a DNA copy of the viral RNA genome. And then that copy, that DNA copy, is copied many, many, many times using something called polymerase chain reaction, or PCR. If you want to drop into the chat, if you've ever conducted PCR before, Dr. Krakowiak loves PCR and will be excited if you've had the opportunity to do your own PCR. There is another test that Dr. Krakowiak and I were quite excited to see. It's called DETECTR, and it's a new CRISPR-based test. This is a rapid test, one of those that you can have the results in less than an hour. And the way that it works is CRISPR-Cas9 is used. And I'm not going to go into how CRISPR-Cas9 works, because Dr. Krakowiak is going to cover that in great detail in her research review next week. But basically, there's this molecular machinery right here. If SARS-CoV-2 genes are present, remember that's the virus, CRISPR-Cas9's action against it, when it cuts it, will start colored on the test strip. DETECTR has 0% false positives and only 5% false negatives, but there were only 90 people in this study, or 90 tests in this study. As we talk about experimental design, when we're thinking about a test that's going to be used to diagnose a bunch of people, I'd hope that there'd be a lot more than 80, a sample size of 80. So that's fairly weak in my opinion. It does require higher levels of virus to show up positive than the RT-PCR tests do. And so someone who has like a low level of infection or an asymptomatic infection with a lower viral load could definitely get a negative test from this when they are in fact infected. But it's been approved for emergency use, so it is out there on the market being used. Now I want to switch over to talking about the antibody test. That's to answer the question, were you previously infected? This right here just shows a lot of different functions that antibodies do. Remember, we've all heard about antibodies, even before COVID-19. Antibodies, another word for them is immunoglobulins. These are proteins. They're very large proteins that do a variety of things. They can bind to receptors to keep a virus from entering the cell. They can keep a virus from being able to uncoat once it has been in the cell. They can even clump viruses together, making it easier for immune system cells to find these aggregated clumps of virus and get rid of them. So the antibodies do quite a lot of things. So how do these antibody tests work? Well, researchers take this lab-grown COVID-19 antigen. So they've made a lot of COVID-19 in a research lab. They have taken antigen. Antigen is just a part of the virus that the immune system can recognize. And usually it's these spike proteins. These spike proteins, the immune system is pretty good at finding them, mainly because they're stuck outside on the outside of the virus, and so they're kind of there for all to see. But the researchers take those spike proteins, put them in these wells, and like immobilize them so they can't move. And then they get some, they extract the part of the blood that has the antibodies from a person and put it in there to see if the person has any COVID-19 antibodies. And this is how it works here. If you have COVID-19 antibodies that are present, they'll stick to the antigens that have been immobilized on this plate. And then there's a detector antibody. The detector antibody just sticks to another antibody. So if a person has a COVID-19 antibody, the detector antibody will stick to the person's antibody, and this will cause a reaction with a color change. However, if there is no COVID-19 antibody stuck to the antigen in this plate, then the detector antibody has nothing to bind to, and it gets washed away and there's no color change. So that's how an antibody test works. With the antibody tests, they need to be highly specific, and that means they have to detect antibodies that are only for SARS-CoV-2. Some of you might have heard that many of us have been infected by other coronaviruses. There are 7 known coronaviruses that infect humans. There's SARS-CoV, which caused the huge SARS outbreak in China and other countries in 2003. There's MERS, Middle Eastern Respiratory Syndrome, which comes from camels, which has infected like 2,400 people and killed like 800 of them. It's a really lethal one. Those are not common. They do infect humans, but they're not common in the population. But the other four coronaviruses, they just cause common colds. And most of us have been infected with at least one of them, if not more of them, in our lifetimes easily. And so with these antibody tests, there's some concern that they could detect antibodies to other coronaviruses, giving the person a false sense of security. So you need a test that is very specific, and you also need a test that is very sensitive. That means that it needs to be able to recognize the antibodies that are there, IgM antibodies and IgG antibodies. These are different kinds. And that way, that the test is not going to give you a false negative. So it needs to be sensitive enough to recognize what's there. The antibodies start developing within 1 to 3 weeks after infection. And so you could have an infection and still have a negative antibody test. I mentioned that earlier. The current recommendation is that even a person with a positive antibody test should still wear PPE. They should still socially distance. They should still stay home when they can. And that's because even if they have a positive antibody test, it's possible it could be a false positive, and their antibodies that were detected were for another coronavirus, not SARS-CoV-2. And it's also possible that a person can have antibodies, but those antibodies could wane over the course of a few weeks or a few months, and then that person may no longer have antibodies to protect them. So that's a concern right now as well. There are over 100 antibody tests on the market. Only about 7 are approved by the FDA. That was back in June. I'm not sure if more have been added to the list. But even among the FDA-approved tests, you can see that some of them are not as sensitive or specific as you might like. So you can certainly take an antibody test and get a false positive or a false negative. And this can lead to a false sense of security, like I mentioned. Actually, if any of you ever donate blood, I get emails from the Arkansas Blood Institute asking me to donate blood on a regular basis. And instead of giving away t-shirts, lately they've been giving away antibody tests. So if you read the fine print, it's an antibody test that has not been approved by the FDA. And I have been unable to figure out the sensitivity and specificity of the test that they are offering. So here's a question for you guys in Pear Deck. Have you been tested? Was it a current infection or an antibody test or both? Or no, you've not been tested? Okay, I'm going to go ahead and show responses, but you can keep adding yours if you want to. So we've had a few people tested, mainly for current infection. 2 people have had an antibody test as well, but the majority of us have not been tested for COVID-19. This kind of makes me sad because I feel like we should all have access to more testing, but this is really just the the situation that we're in right now. Okay. So we're going to move on now to the search for treatments. So I'm going to be focusing on antiviral drugs and also treatments for symptoms of COVID-19. There are a few places online where you can track the treatments and vaccines that are being studied. This is one right here. The milkeninstitute.org has a COVID-19 tracker that right now they're looking at 271 different treatments and 198 vaccines, and they are keeping, it's a way that you can look at the different treatments and vaccines and link to the different sources about these. But even maybe better than this one, I really, really, really like this one. And I found this one just a few days ago. This is put out by the New York Times. They're following a few different coronavirus treatments, and they rate them as being promising evidence, sensitive or mixed evidence, not promising, or pseudoscience or fraud. And I'm going to be showing you guys some screenshots from this coronavirus treatment tracker as we go through. The URL is down here if it's something that you want to open up and save and bookmark for yourself. So first, I'm just going to pull their 3 examples of pseudoscience, fraud. Don't do these things. I also want to mention, I know I said this morning, don't have walls of text, and please, please don't have walls of text, because you guys want to read this now. You want to read all these paragraphs. I know that you do. I tried to put the important stuff in yellow, but I wanted to show you guys what this treatment tracker is like so you are able to use it. So things like drinking or injecting bleach or disinfectants, this has been suggested by people on national TV. Please ignore this suggestion, it is dangerous. There's certainly no evidence for UV light either, which has also been speculated about. UV light can sterilize surfaces. It cannot purge virus from inside a person's body. What it can do is give you skin cancer. Also no evidence for silver. There are some pretty prominent names who are hawking products claiming they are effective against COVID-19. There's no evidence for this, and they are scams. So with that in mind, can you guys, using Pear Deck, name one specific COVID-19 treatment? Try to give me the name of the medicine or the drug, something that's being used or studied right now. You can also say IDK or I don't know. Good, I see, I do see a lot. Right now I see 2. Okay, I'm gonna go ahead and, well, I'll give you guys a couple, I know some of you are still typing a little bit. I'll give you another few seconds. Yeah, I see 2 coming up over and over again. And one is a drug with evidence behind it, and one is a drug with no evidence really to support it. So we'll look at that here in a minute. Let me show your responses. We're looking at remdesivir, remdesivir, lots of remdesivir, that's great, and hydroxychloroquine. Hydroxychloroquine has had numerous research studies behind it, and as we will see, I'm not sure what Japanese flu medicine is. That's, I'm not sure which one that one's referring to. Hydroxychloroquine, though, ended up pretty much failing its clinical trials, but you are correct that it was being studied in great detail. So let's kind of go on for a minute. This right here is from a Scientific American article, and we should give this to you. So we need to drop the Scientific American excerpt because it's a really, really good read into your student binders, and we'll try to remember to do that when I put my slides in later. But this right here is showing you that there are basically 3 ways to treat COVID-19. There are different ways, like remdesivir right here, that block viral replication. There are other ways, like different kinds of antibody candidates that prevent entry into the cell. And then there are treatments that are aimed at helping with the symptoms, and in particular, reducing a hyperimmune response. And we'll talk more about that hyperimmune response here in a second. But this right here is why COVID-19 can be deadly. It can cause major cytokine storms, huge overreactions of the immune system, major systemic inflammation, organ failure, etc., and acute respiratory distress because the immune response is really overinflated. And these right here are aimed at reducing that overaction by the immune system. So I'm going to talk about that first one, prevent viral entry, and then we'll go on to the other two. Right here, you see a pretty cool figure where we've got SARS-CoV-2, remember that's the virus, entering cells by its receptor. So here the orange is the spike proteins on SARS-CoV-2, and they bind to a receptor that's shown here in blue on a host cell, and that receptor is called the ACE2 receptor. ACE2 stands for angiotensin converting enzyme 2, and it's a receptor that plays a role in other physiological processes. As a side note, SARS-CoV, which caused the SARS epidemic in 2003, that also uses ACE2, but at less than 1/10 of the affinity. So SARS-CoV-2, the current virus causing COVID-19, it's more than 10 times better at binding this receptor to get into cells than that SARS outbreak of 2003. So just to kind of let you know what's going on here, this right here is a normal physiological process. So ignore the virus for a minute. You've got some things called angiotensin 1 and angiotensin 2, and they've got a few different receptors. This one right here, angiotensin 2 type 1 receptor, if it's being activated too much, it ends up resulting in lung injury, inflammation, and leaky vessels. ACE2, this is the one the virus uses, it actually breaks down angiotensin 2 to stop angiotensin 2 from causing this lung injury and inflammation. However, when the virus, SARS-CoV-2, binds up all of this ACE2, there's no longer any regulation of this other pathway, and that's why you can get some runaway inflammation happening in the most severe cases of COVID-19. So that's basically what all this says. When we have less ACE2, because the virus is using a lot of it to enter the cells, we end up getting way too much of this other pathway that's normally held in check over here. But knowing that this is how the virus gets into the cells gives us some ideas for treatments and symptoms. So there are various types of ACE inhibitor drugs and ARB drugs that are aimed at preventing this inflammation pathway from happening. There's even kind of a cool idea, I think, a synthetic decoy molecule, APN01. This is basically a synthetic ACE2 version that is meant to attract coronaviruses to the synthetic molecule and then trap them so that the virus doesn't bind the real ACE2 receptor. So that's something that has been studied. This is from that treatment tracker that I showed you guys earlier. And that recombinant ACE2, having some of this that's just synthetic, like injected into somebody to try to trap the virus, has tentative or mixed evidence right now. So promising results in cells, but studies in animals and people are still ongoing. So this one right here is tentative right now. Now we're going to talk about antibodies. So the antibodies, what happens here, we know that antibodies protect us from pathogens. So you can have a person who has been previously exposed, they contracted this coronavirus, they fought off the coronavirus, and they now have antibodies, protective antibodies circulating in their blood. They can donate that to be transferred into a current COVID-19 patient who needs treatment. And so this is called convalescent therapy or passive antibody therapy. Because a donor can only give enough for maybe 2 to 4 other people, and there aren't enough donors right now, as we'll see in a second, there are also lab-developed antibodies called monoclonal antibodies that are also being studied. And the nice thing about the antibodies is that they can be given in a variety of circumstances. They can be given to like frontline healthcare workers, employees in nursing homes, and other essential workers on like a prophylactic basis, basically pre-exposure to try to keep someone from getting sick. They could also be prophylactic post-exposure, meaning people that have been identified by contact tracing, particularly those that have high risk factors, they could be given these antibodies to help them hopefully keep from developing COVID-19. They could also be therapeutic. My cat has just jumped onto my table, meaning for people who are already ill. And this is how they're currently being used, in large part because we don't have enough of them to do these other routes of administration. So right now, what antibodies we are collecting are going into those people who are very seriously ill. UAMS actually has FDA approval for emergency use of convalescent antibody therapy. If you know anybody who has been treated with convalescent antibody therapy and you want to share that in the chat, you can do that. And this is what the treatment tracker says about convalescent plasma. This is one that would be donated by somebody. And then monoclonal antibodies, those are the ones being made in the lab. Again, we kind of have promising but preliminary results that they work. Safety trials for the monoclonal antibodies are only just begun, so that is still kind of in the research pipeline.
While you're on antibodies, can I ask a question that Victoria had? She's
Sure.
saying, don't antibodies primarily work to alert the immune system, but don't actively work against the virus? And this has to do with the binding and so forth. So maybe
yes, they bind to those spike proteins. So antibodies have two ends. They have like two hands on one end to grab on to spike proteins, and then they've got a tail on the other end the immune system uses to recognize that the antibody is there and bound to something. And so these antibodies, they can bind to the spike proteins, help the immune system find that the virus is there. But also, if you have all those spike proteins coded in antibody, they can't bind to ACE2. So they can't get into a host cell. The antibodies basically block them from being able to get into the host cell if you have enough antibodies that are able to bind to those spike proteins on the virus. That's a good question. I do want to say, starting as of this past Monday, the Arkansas Blood Institute is entering people in a giveaway for thousands of dollars if you can donate convalescent plasma. So if you know someone who has tested positive for COVID-19, for SARS-CoV-2, they've had this coronavirus, they have recovered from it, they have antibodies to it in their blood, please encourage them to give blood. Please, please, please. The Arkansas Blood Institute says right here, the need for convalescent plasmid in our area has increased over 700%. Demand now regularly greater than collections. They only have about 20 people coming in each day to donate convalescent plasma. That's why they're starting to give away $1,000 twice a day to donors. If you know someone who can have these protective antibodies, please ask them to donate blood. They can really help some people. So here's a Pear Deck question. Do you know anybody who has tested positive for COVID-19? You don't have to tell us who, since that's somebody else's medical history, but we'd like to know if you know someone who's tested positive or not. I only know someone who knows someone. I don't know anybody myself. I'm gonna go ahead and show the responses. Looks like it's about half and half, tending a little bit more towards, yes, I know someone who has tested positive in the past. Again, ask them to donate. They can win some money and they can help some people. Hide those responses and go on.
Another question, somebody says, don't antibodies only last 3 months or at least for COVID? I don't think we know yet, but.
We don't really know yet. Some antibodies can last. So antibodies themselves typically break down in less than a year. Someone who has recovered will also usually have memory B cells and memory T cells, which are capable of turning on an immune response if the person is reinfected, and that will create more antibodies. The initial antibodies do tend to wane over time. It does depend on the pathogen. And for SARS-CoV-2, we just don't know yet because it's so new. We don't know if these are antibodies that are going to last for a year or 2 or 10 years, or if these are going to be antibodies that are kind of gone in 3 or 4 months. And that's an open question. We all hope they last long time. So now I'll talk about blocking viral replication. This is an image of the of the virus's replication cycle. So you can see the antibodies right here. They try to block this from happening. So this is the ACE2 receptor. They try to block the SARS-CoV-2 from being able to bind to its receptor. Chloroquine and hydroxychloroquine were thought to potentially help, this is that anti-malarial drug, help keep the virus from being able to get out of this endosome, but it turns out they're not terribly effective. Remdesivir is another big one that's over here, and it actually is blocking viral replication. But the key here is that the scientists have to know this. In order to find treatment candidates to study, scientists have to know how this virus replicates in order to be able to interfere with specific steps of that process. So we're going to start out here by talking about remdesivir. Remdesivir interferes with the RNA polymerase. It keeps it from being able to replicate its genome. There was a clinical study. It was randomized. Remember how good that is. So randomly assigned over 1,000 COVID-19 patients in multiple countries. That's good too, because that means hopefully they weren't just studying 1,000 white males, right? Hopefully we had some diversity there, people from different genetic backgrounds, to get intravenous infusions of remdesivir or a placebo. So here we have randomized, placebo, controlled. I don't remember if it was blinded or not. Remember, if it's a double blind, then I don't believe it would have been blinded. I think that they knew who they were giving the remdesivir to and who they were giving placebo to. But still a very strong study. You know, over 1,000 people, randomized, placebo controlled. That's not as good as having the double blind in there too, but that's pretty solid. The mean time to recovery was shorter. Okay, guys, the people who got remdesivir, they were discharged from the hospital 4 days faster, 11 days versus 15 days. Here is that p-value. We've talked about p-values. They use, you know, a t-test to assess these people, to assess the two groups, and the p-value is less than 0.001. That's great. That means significant because it's less than 0.05. Now, when remdesivir, when the results first came out, preliminarily, before they had all of the data, they said that mortality looked like it could be reduced, but the p-value was only 0.07. 0.07 is close to being less than 0.05, but it's not quite there. However, now that that study has concluded and they have outcomes for all 1,000 of these people, we now see that mortality in the remdesivir group was 7.6%. It was 12.5% among the patients that did not get remdesivir, and that is a significant p-value. So that is really good news regarding remdesivir. Of course, even in the remdesivir group, almost 8% of the people still died. And so it is not a magic bullet, but it is nice to have in our arsenal. There have been some other stuff with remdesivir. They've looked at pediatric patients. Of course, that means children, only 77, so that's pretty weak. But they did find that 73% were released from the hospital by day 28. Only 4% died. So it seemed like remdesivir hopefully helped a little bit in some of those cases. They've also looked at infected pregnant women with remdesivir and found that remdesivir helped lessen the amount of extra oxygen needed in 96% of pregnant women and 89% of women who had newly given birth. So you've got women who are pregnant or have just delivered in the hospital who are infected. Remdesivir has helped reduce the amount of extra oxygen that they needed. That's another good thing. This is what that treatment tracker says about remdesivir. This is what they said. The early results did not show any effect on mortality. That was that 0.07 p-value. But the retrospective data, when that was completed, said that it does look like there is a reduction in death rates among those who are very ill. So that's remdesivir. Now there's hydroxychloroquine. So this one was promising initially. I mean, it did inhibit SARS-CoV-2, that's the virus, in monkey cells. It helped, again, so in like a test tube, monkey cells in a test tube, they were able to inhibit SARS-CoV-2 in that situation. But when it entered large trials, they found that it was no better than placebo. So they had looked at healthcare workers, 800 people. This was randomized and double blind. That's very important. Remember, that means that the healthcare workers and the researchers giving them the pill did not know if they were giving a placebo or hydroxychloroquine. So it was randomized, double blind, 14% versus 12%. The p-value was greater than 0.05. There was a little bit of a caveat in that they determined who was sick based on symptoms, not by an RT-PCR test. So these groups may be missing asymptomatic cases. And that is a potential weakness here. But ultimately, there were several follow-up studies, and in the end, the FDA canceled the emergency use of hydroxychloroquine, and the World Health Organization stopped testing it. I'm going to go, can I get my slides to switch? Okay, this is what the treatment tracker says, not promising. I also want to point out, I meant to say this back here, it's known to interfere with heart rhythms and can lead to sudden heart attacks, and it's currently being used to treat people with these, with lupus and rheumatoid arthritis. These are people that live in a great deal of pain with really horrible symptoms every day, and they are under the care of a doctor, and their hearts have been checked out by cardiologists, and so they have been cleared to take this drug that does things that can have ill effects on the heart. With the drug's newfound publicity, there was a lot of demand spike. There were at least 6 states that had to increase their regulations of prescription hydroxychloroquine because a lot of prescriptions were being written improperly. And there were actually shortages for people who rely on this for treatment for other diseases, which I think is very unfortunate. But in the end, there were various clinical trials, and none of them really panned out. And the FDA and the World Health Organization have all said that there's really no reason to continue studying hydroxychloroquine. Some of you, I don't know if it's coming up in the chat box, I had a family member a few days ago tell me that a recent study has proven that hydroxychloroquine works. It did not. If you have seen something about the Henry Ford Foundation's hydroxychloroquine study, they made a fundamental flaw in their experimental design, in that they have more than one difference between the groups. So they had 2 groups. They had their hydroxychloroquine group and the group that did not get hydroxychloroquine. Unfortunately, the group that got hydroxychloroquine was also twice as likely to get a steroid treatment that is also known to help reduce mortality in COVID-19. And we'll talk about that steroid here in a minute. So you can't give a group hydroxychloroquine and a steroid and then say it's only hydroxychloroquine that is causing the outcome, because you've given them 2 different things. I'll also say that the Henry Ford hydroxychloroquine trial was not randomized. They looked at different patients. They chose the patients they thought would do better on hydroxychloroquine and put them in the hydroxychloroquine group. This is not how you do science. Now, is it still possible that hydroxychloroquine could combine with another treatment to be beneficial? Yes, that's within the realm of possibility, but we need scientific evidence for that. You know, we need well-designed clinical trials, and we don't have that right now. So I'm now going to go on to number 3 of that list. That's how to treat the most lethal symptoms. Cytokine release syndrome, or CRS, is what happens when you've got this really overactive immune response with interleukin-6. This is an immune system signaling compound. And you end up getting this. You get people with mucus-like fluid buildup. They get ARDS, that's acute respiratory distress syndrome, and they go on ventilators. And these symptoms are the main reason why a lot of people with, the reason that a lot of COVID-19 patients will end up dying is because of this right here, because of this overactive immune response. And so there are a few different possibilities. There's some cytokine inhibitors like IL-6 blockers. These have been studied. Several of them are in clinical trials. There's one, this Kevzara right here. I think that you can't see the results is under the Pear Deck bar, but it has recently failed phase 3 trials. So this one ended up not really working. And so this is kind of mixed evidence for the cytokine inhibitors that are just trying to dampen this overactive immune response. One that there is some evidence for is dexamethasone. If I were going to have a loved one in the hospital with COVID-19, I would hope that they were being treated with remdesivir and dexamethasone. These are the ones that, from the research I have done, seem the most promising at this point. So dexamethasone is the steroid drug. It's anti-inflammatory, it's immunosuppressive. This was the first drug shown to reduce COVID-19 deaths. Remdesivir has since come out following that being the second one. This drug reduced death by 30% for those on ventilators and 20% for those on oxygen. This was in a group of over 6,000 people. This was published in the New England Journal of Medicine, which is a major journal. Dexamethasone, it's inexpensive. It can be taken orally or by the IV. Hospitals already have it in stock. It's called a GRAS drug, G-R-A-S, generally recognized as safe. In 2017, it had over 1 million prescriptions. Like, this is a drug that has been used in humans, used. So it's great that it's helping with our most severe cases of COVID-19. It is not for prophylaxis. That means that you're not supposed to take it to keep from getting COVID-19. Remember what it does. It dampens the immune system. It prevents the immune system from overreacting and creating organ failure, basically. You don't want to be taking it when your immune system is fine. You want your immune system to be strong to hopefully be able to fight off SARS-CoV-2 if you do contract it. And you only want dexamethasone if you have a very severe case. So there's that one. Here's what our treatment tracker says about it being very helpful for patients, over 6,000 people in that New England Journal of Medicine study. So now we're going to spend the last few minutes talking about that race for a vaccine. I want you to choose an option on your Pear Deck. Do you plan to get a COVID-19 when they become available? I'm looking for about 10 more responses. And I know it might be a hard question. You might think, I don't know, I haven't thought about it yet. Here are the responses that we have got right now. So most people saying yes, some people saying no for a couple of reasons, a few others. If you have another reason and you're comfortable sharing it in the chat, you're welcome to do so. Although I do want to say that vaccination can be definitely kind of a hot topic. I want to remind you all about your code of conduct agreement and how we want to speak respectfully and kindly to each other. Let's see. And so first I want to talk just about the basics of a vaccine. This is how we develop immunity. You get an infection, the virus is in your body. Your immune response is aiming to recognize the virus. Typically, the spike proteins are one of the major things that it's able to recognize, make antibodies, B cells, T cells, all working together to eliminate these viruses, and ultimately do this to give long-lived memory B and T cells that are going to patrol in the body for months or hopefully years or hopefully even decades to keep you safe if your body ever re-encounters this virus. That's how we develop immunity. So this is the goal of vaccines, to build memory. So this is a Pear Deck question for you. Drag the red star to the part of the graph that best represents where we want the vaccine to be administered for optimal immune system fighting of a pathogen. While you are dragging the red star around, I'm going to talk about this for a little bit. With immunity, you have your initial exposure. Initially, you can see that the y-axis here is concentration of antibody. Initially, there's really not any antibody. It takes, a week or two, and then the body starts to make some antibodies. They tend to be low affinity, meaning that they recognize the virus, but maybe not that well. And then later on, you have a second exposure, and look, there's no lag time. Here, we just jump right up to the top. We get antibodies very, very quickly, even more than the first time, and they're higher affinity antibodies. So I am now going to lock screens and share response. And this right here, these right here, this is where we want where we want the vaccine to be. Because think about it like this. We want the vaccine to be the initial exposure. The vaccine, it's been studied in clinical trials. It's been deemed safe. We want to get the vaccine to have a week there for our body to be learning how to recognize that virus and making those low affinity antibodies and making those memory cells, all based on the vaccine, so that the secondary exposure, so that if we get exposed to SARS-CoV-2 in real life and that virus that causes COVID-19 gets in our body, we don't want to have to do this. We don't want it to take a week for our body to learn how to recognize it and make some low affinity antibodies. We want our antibody titer to skyrocket and make high affinity antibodies, which it is prepared to do if it's already seen the virus previously in a vaccine. So that is the goal of vaccines, is to build that memory first, so that the first time you encounter the real deal, it's actually your second exposure and you can have a memory response instead of this initial response. So very good there. I want to talk for a couple of minutes about what does acceleration actually entail, because I think that the fact that this is an accelerated vaccine pace worries people. And so what does that mean? Well, first of all, a lot of steps are being performed in parallel. In traditional vaccine development, you can spend months or years on phase 1 and then phase 2 and then phase 3. The manufacturing process doesn't really start immediately. And even when you're in phase 2, you're just doing small batches for trials. If phase 2 is going well, you start to scale up and validate in the manufacturing process. And the large scale manufacturing, it doesn't even start until the approval and licensing has taken place. Whereas once you are in the pandemic pace, that's what we're kind of in right now, we have things that are happening in parallel. So some phase 1 and phase 2 are being combined, some phase 2 and phase 3 are being combined. We had phase 1 starting before all of the animal tests were even done. And really right here is what I think is fantastic, is the manufacturing process. Look, they're scaling up and validating. They're getting ready to make massive amounts of these vaccines before we even know if they work. And that's so once we know which ones work, the manufacturing process has already been figured out. So we're already getting large scale manufacturing and emergency authorization ready before these things have even been completed. And that's so when we say, yes, these are safe, we don't have months of manufacturing to work out. Another thing is more money. The United States has just offered to pay, or has agreed to pay, $1.6 billion to Novavax for a coronavirus vaccine. Novavax is one company, and they're not even the only company that's getting money from the government to make vaccines. So there is so much money being pumped into this right now. And that's one of the things that helps it to speed up, is because you don't have scientists begging and applying for grants and trying to get this stuff through, and can take years to get those grants because the funding is being made readily available. I go to another slide here. I also am going to mention, let me make sure I didn't skip something. Okay, so we know that traditional vaccines often use debilitated viruses or purifying proteins, and it can be difficult to make enough of that for millions of people. Like, it can take years to make enough viruses of traditional vaccines. And that's why we are mainly using genetic engineering and virtual modeling. And so, for example, for one vaccine, they took the virus's RNA, they converted it into DNA. You can do that easy, like on a computer, it takes 2 minutes, you know. They use simulations to find the best regions to simulate antibody production. It took 3 hours to design this vaccine, and it took a month to manufacture. And this is a big reason why it's possible to do this without compromising safety, because these are brand new tools. The genetic engineering and the virtual modeling, this is something that's only become possible in the last few years. And a lot of it has been kind of figured out in the search for a vaccine for Ebola and Zika. And so the groundwork has already been laid, and we have this brand new modern technology to help us do it. At least 30 vaccine trials were already testing.
There's some questions about safety, and I think that's what's been talked about right now, that there are these new technologies being used, and talk about how those are not the safety concerns.
Oh, those are definitely not the safety concerns. Basically, instead of it taking scientists years to figure this out in like a wet lab, they can use simulations to figure out what they need to know, and then they can test it in a couple of weeks. And there's no safety concern there. It's amazing that we can do this. We can use computers, and we can use math, and we can use computer science to design these programs that give us a better understanding of these viral surfaces and the molecular interactions between viral spike proteins and human cells and human antibodies, things that we have never been able to figure out before. And so it's really, really good that we have this kind of technology. And this right here is not a problem with safety at all. These are some other notes on acceleration and safety from experts. They're talking about how we now know how to measure the immune response with much greater accuracy in humans. We have cleaner and likely safer technologies to create the vaccines. We know the viral proteins that are likely to raise a good immune response. We know how to measure that immune response better than we've ever known how to do it before. There's also more openness and sharing. This is Dr. Margaret Hamburg from the National Academy of Medicine. And she says that when people were looking for a vaccine for Ebola or Zika or H1N1, everyone was trying to get there first. Nobody was sharing with anybody. It was super secretive. And now we've got regulatory authorities from all around the world who are reducing barriers, bringing people together, giving them all the funding they need to make this happen, to work together. Here's another great example here. This Dr. Susan Weiss says our CDC permit to receive the virus. Remember that this virus has to be, it's dangerous, it can spread from human to human, you can't let it escape out of a lab. So it takes only certain labs with certain approval can get this virus to work with. And she says that her lab was approved in less than 2 days. She received at least 2 material transfer agreements, which have to be signed by a number of officials and lawyers in a matter of hours. Those things sometimes take weeks in the past because the form will just be sitting on somebody's desk while they're on vacation, you know? And now, because it's so important, everybody is able to say that yes, the Penn Center for Research on Coronaviruses and Other Emerging Pathogens that has been studying coronaviruses for years, we're going to approve them to get this virus and begin the work, and we're going to approve them today. It's not going to take a month to get signatures on a piece of paper. That's another big part of the acceleration right there. Having a hard time switching slides here. This is, a lot of this is in the Scientific American article that we're going to share with you guys, but it goes through the pros and cons of the different kinds of vaccines. So we've got DNA vaccines, we've got RNA vaccines, an mRNA vaccine tested in, this is with a phase 1 trial, so only 8 people, to make as many or more antibodies as those who had recovered from actual COVID-19. So this is an example of one of these nucleic acid vaccines. We've also got viral vector vaccines. This is where you have a weakened viral vector. A non-replicating one like adenovirus is the most common one. Adenoviruses cause common colds. We've all been infected with an adenovirus. It's not a big deal. But they can be genetically engineered where instead of causing a common cold, they're actually carrying some SARS-CoV-2 DNA. And that means that some coronavirus proteins are expressed, and the immune system can then learn to recognize those coronavirus proteins that are being expressed, the SARS-CoV-2 proteins that are being expressed, even though you're not actually infected with SARS-CoV-2, right? None of its other genes are there, just the genes for those proteins on the outside. There are also protein-based vaccines, sometimes called subunit vaccines. This is where proteins like the spike protein, that's the protein that the virus uses to get into a host cell, those can be injected directly into the body or as part of something called virus-like particles to get an immune response that can recognize those spike proteins before the actual virus is ever encountered. Now this is a different tracker. I showed you earlier the treatment tracker. This is the coronavirus vaccine tracker. Since we're coming up to the end of time, I'm not actually going to go to it, but I noticed these are the numbers from 4 days ago, but then I checked it at lunch today. Just since 4 days ago, there are 4 more vaccines that are preclinical. There are 3 more in phase 1 and 1 more in phase 2 or something like that. So we have movement. We're moving these vaccines through and they're being tested. And you might notice this right here. I'll put the link to the coronavirus vaccine tracker into the chat a little bit later. It's also down here under the Pear Deck bar, but it's also at the New York Times. You might notice that you've got one right here approved for limited use. Maybe that's a bit of a surprise that already one is approved. And it's this one, CanSino Bio, has limited approval. This is a Chinese company, and the Chinese military has approved this vaccine for soldiers, so specifically for the Chinese military. I couldn't find any evidence that any soldiers had actually been vaccinated with it yet. I don't know if vaccinations are actually happening, but at least it has been approved for use in the Chinese military. Currently, that's the only one approved for sort of wide use in humans.
There's a quick question, and I'll forget. What BSL level is COVID, and has it been increased recently?
I assume it's BSL 3. I think that the coronavirus lab at Vanderbilt, where I used to work, was a BSL 2, but it might need BSL 3 for this one. That's a very good question, and I would have to look that up. But I'm sure it's Googleable. Like, I mean, you can probably do SARS-CoV-2 BSL and figure that out. Maybe somebody can put that in the chat, and then I'll know for sure. This is a series of polls for the percent of Americans who say they will get vaccinated. This one was almost 11,000 people. There were a few other polls of 1,000 people each that were also in this kind of 50 to 70% range. And I think that you can kind of see that it depends a little bit on one's politics and background. I want to know, though, I want to sort of ask you guys again in Pear Deck, did my talk of what the vaccine acceleration actually means, did it change your mind? And if you said no before, do you now say yes? If you said yes before, do you now say no? I'd like to know if you still feel like you would get, you can still get the vaccine. My hope would be that I've convinced you that yes, you don't have to be afraid of this vaccine. A large part of the acceleration is just about already starting the manufacturing and having the money to do this. I do not believe that safety is being compromised. Let's see, I'm gonna let maybe 10 more people give their responses. And then there's just a handful of slides after this one. I'll go ahead and show the responses. I think that we may have had a bit of a shift towards yes, but I'd have to go back and look at the results from the other poll first. But there we go. I'm gonna hide those responses. This is just some links for further study if you guys are interested in this. The coronavirus vaccine tracker is this one right here. I forgot to add the treatment tracker right here. There are also some books. If you're a podcast listener, there are a few different podcasts that have addressed the coronavirus pandemic that you might be interested in listening to. Also, I recommend Science News. Science News, there's a lot of different articles that I recommend that you can check out if you're interested. But I also think if you are willing to get a special newsletter, you can go to sciencenews.org/special-newsletter. I get this newsletter once a week. It used to be twice a week, but they've dropped it back to once a week. And it is such a great summary of the most recent research. They link to actual research papers. They talk about what the actual findings are from studies, and I think it's a really great one if you'd like to be keeping up with that. And then I did make my slides from slidesgo.com. We talked about that one earlier. I had a few photographs from freepik.com. And then these are some of the other peer-reviewed, mostly peer-reviewed sources that I use. We do have where was that Scientific American? Right here, the Vaccine Quest, Scientific American. we'll put that one in your folder. Oops. And so thank you very much. I know that it's slightly after 4. I think that I did a pretty good job staying to an hour, and I'm happy to take any questions.
Guys,